Good evening, this is Mr. Wintermelon. In this video, you will learn the second form of cell division, and the name is called meiosis, or what I like to call 没有事,没有事,没有事, because this is what your parents is going to tell you when you step into their bedroom at the wrong time. So by the end of this lesson, you should be able to identify with the aid of diagrams the main stages of meiosis, as well as state how meiosis and fertilization can lead to variation of the offspring. And again, I would like to emphasize that description is not even secondary. It's really, you do not need to write essay on the stages of meiosis. You just need to point out that, oh, this picture it is which stage of meiosis. The only difference is meiosis, there are more parts as compared to mitosis. As mentioned, meiosis consists of more parts than mitosis. Therefore, we have meiosis 1 or meiosis part 1 and meiosis 2. During meiosis 1, the homologous chromosome will separate. So because the homologous chromosome separate, the cell will be considered as haploid, as they only have one set of chromosome left. The so-called sister chromatids that are still inside, we kind of consider them to be identical. So they are just left with one set of chromosomes. They can be 10 from the father, 13 from the mother, or it can be 6 from the father and 17 from the mother, whichever the permutation or combination allows. In meiosis part 2, each of the daughter cell further divides itself, so we will end up with 4 daughter cells. And in meiosis part 2, the sister chromatids will separate. So meiosis part 2 is very similar to mitosis. The difference is the daughter cells will end up with 23 chromosomes instead of your 46. So let us look at the video describing meiosis. For meiosis, there is actually meiosis part 1 and meiosis part 2. For the prophase of meiosis part 1, it is actually more interesting than the prophase of mitosis. So we can see that the homologous chromosome, they are paired up. So the paired up homologous chromosome, they are undergoing a process known as crossing over, whereby one of the sister chromatids will exchange the genetic material with the other. And the end result will be something like this. So by the end of prophase 1, crossing over will have occurred, and you can see that the homologous chromosome, they will be genetically different already, because part of the mother chromosome have been transferred to the father chromosome, while part of the father chromosome have been transferred to the mother chromosome. So this is important to help give rise to genetic variation in the daughter cell. After the end of prophase 1, we will start our metaphase again, but it is different from the metaphase of mitosis, because for meiosis, the metaphase 1 is the homologous chromosome that will be aligned in the middle. So just recall, for mitosis, the metaphase is the alignment of sister chromatid. But for meiosis, it's the aligning of the homologous chromosome. So there is a difference. So your homologous chromosome, they will align themselves or meet up in the middle during metaphase. And how they are arranged, it can be like this or it can be like this. This is known as independent assortment of chromosome. This is important because it will also give rise to genetic variation in the daughter cell. Because you can imagine, if it's like this, compared to like this, the daughter cell will receive different homologous chromosome. During anaphase 1 of meiosis, the homologous chromosome will get put to the opposite end. This is different from mitosis because for mitosis, the anaphase, the sister chromatic will get separated instead of the homologous chromosome. So for mitosis, anaphase part 1, the homologous chromosome will get separated to the opposite end of the cell. So centriole will pull the chromosome to the end, and after anaphase 1, it will be telophase 1. So during the telophase 1, the cell will officially divide, and the resulting daughter cell will be genetically different from each other, because you can see that, oh, this daughter cell got the shortest chromosome from the father, while this daughter cell have the shortest chromosome from the mother. So since they are genetically different already, they only have one set of chromosomes. Unlike just now, they have two sets. Now they are considered haploid. So by the end of meiosis part 1, the daughter cell are haploid. And we will continue our cell division with just one cell. Later on, I will bring in the rest. Now we start with meiosis part 2, beginning with prophase 2. It will be the same sequence: prophase 2, metaphase 2, anaphase 2, and then telophase. So similar to mitosis, the prophase 2 of meiosis is pretty boring. Those so-called sister chromatic will just be down there, not in the middle, minding their own business like pedestrian. After prophase, it will be metaphase. So we have our centriole that will align the sister chromatid in the middle. So this alignment is another round of independent adsorption. By now you should be able to see that depending on the orientation of the chromosome, the daughter cell will get either this sister chromatid or this sister chromatid. So the so-called sister chromatid, they will be aligned in the middle or meet up in the middle during metaphase 2, or what I like to call metaphase 2. Anaphase 2 of meiosis is similar to anaphase of mitosis because the sister chromatid will separate to the opposite end. So the centriole will pull the sister chromatid or the chromosome to the opposite end, and after anaphase 2, it will be telophase 2. So the cell will divide into two through cytokinesis, and the resulting daughter cell, you can see that they are genetically different. These two chromosomes, they are different. This one, even though they are the same, but the longest chromosome they are genetically different due to crossing over. So we have one set of chromosome down here, and another daughter cell can have this set of chromosome, and the last one can have this set of chromosome, or it can just have like this. It is also possible. You can see that all of them are genetically different, and this is meiosis. It will give rise to genetically different offspring due to crossing over as well as independent assortment. And as a result, at the start, the cell have 6 chromosomes, but the daughter cell, okay, let me just take out the cell. We will end up with 4 daughter cells, and each of them will only have 3 chromosomes because of 2 cell division that occurs. Unlike mitosis, whereby the daughter cell have equal amount of chromosome as the parent cell, for meiosis, the daughter cell will have half the original number. And each of the daughter cell, they are genetically different. So this is just an example from a cell with 3 pairs of chromosomes. Imagine human, we have 23 pairs of chromosomes. The permutation and combination will give rise to even more variety, which is why we are so different from our parents. Yes, there are similarities, but overall, most of the people can tell you and your parents apart. Unlike the prophase of mitosis, the prophase of meiosis is actually pretty interesting. Here we have some description of prophase 1, which honestly, I would say don't bother, because the more important thing is you need to be able to identify prophase 1 from the diagram. And if there's anything that you need to copy down, it is this. Crossing over takes place during prophase 1 of meiosis 1. Crossing over is an exchange of genetic material between homologous chromosomes. This means part of the father chromosome might swap with part of the mother homologous chromosome. This will produce new combination of alleles or new combination of genes. That means originally this gene might pair up with one gene, but after crossing over, it might pair up with another form of the original gene. This is very important. Crossing over during prophase 1 can give rise to genetic variation. After prophase 1 is metaphase 1 or metaphase 1. Similar to metaphase of mitosis, you need to be able to identify that the genetic material are in the middle. But unlike mitosis, whereby the sister chromatic align themselves, during metaphase 1 of meiosis, the homologous chromosome arrange themselves in the middle. And this is known as independent assortment of homologous chromosome. And this will also give rise to genetic variation, similar to crossing over. This is important. You need to take note that during metaphase, independent assortment of chromosome will give rise to genetic variation. Each chromosome is attached to spindle fiber. This one all of us know. And I will use the example of sex chromosome in the slide to illustrate the separation of chromosome. So similar to mitosis, the DNA will also have replicated. So you will end up with 2 X chromosome and 2 Y chromosome. The 2 X chromosome are the sister chromatics, while the X and the Y, these 2 pair are the homologous chromosomes. I am using the example of sex chromosome because it is easier for you to see. After metaphase 1 is anaphase 1. How creative is the name? During anaphase 1 of meiosis, the homologous chromosome, they will get separated, being put to the opposite end by the spindle fibers. So the difference from mitosis is the homologous chromosome actually get separated. Unlike mitosis, whereby the sister chromatic get separated. And this is why we will end up with haploid cell. After anaphase 1 is telophase 1 or telophase 1. You need to be able to identify the cell is in telophase, which is not difficult because the chromosomes are at both ends. And cytokinesis will take place, giving rise to 2 haploid daughter cells. After meiosis 1 have finished, we will go into meiosis 2. And the first stage of meiosis 2 is prophase 2. Prophase 2 is pretty boring, like the prophase of mitosis. Basically, you just have chromosomes that are sitting around minding their own business, and they are like in a mess. So you just have to identify the mess from the diagram and say that, oh, it is prophase 2. And just take note, there is no crossing over that occurs during prophase 2. So after the boring prophase 2 is the more interesting metaphase 2. Similar to other metaphase, the chromosome will align themselves in the middle. But unlike metaphase 1, whereby the homologous chromosome is in the middle, it is the sister chromatic that arrange themselves along the metaphase plane. And just take note, there is some sort of independent assortment of the sister chromatids, because for metaphase 2, the sister chromatids are not exactly identical due to the crossing over which took place in prophase 1. So how the sister chromatic arrange themselves will also give rise to genetic variation. After metaphase 2 is anaphase 2. Similar to other anaphase, the chromosomes are being put away. So when the sister chromatic gets separated, they become individual chromosomes. So for anaphase 2, you will see 4 sets of chromosomes, each at the opposite poles of each of the cell. And finally, we have our telophase 2, whereby the chromosomes are at both ends of each of the cell, and cytokinesis will take place, and the outcome is we have 4 haploid cells that are genetically different from one another. So this is all for the stages of meiosis. There's part 1 and there's part 2. Part 2 is pretty similar to mitosis. So let's compare the metaphase and anaphase of mitosis with metaphase 1 and anaphase 1 of meiosis. First, let us look at the metaphase. For mitosis, during metaphase, the sister chromatids are arranged in the middle. While for metaphase 1 of meiosis, the homologous chromosomes are arranged in the middle. So mitosis is sister chromatids, while meiosis is homologous chromosome. And for anaphase and anaphase 1, for mitosis, what is separated are the sister chromatic, while for meiosis, it is the homologous chromosome that get separated. So this is the difference between metaphase and anaphase of mitosis versus the metaphase 1 and anaphase 1 of meiosis. Lastly, I have a table comparing the difference between mitosis and meiosis. So for mitosis, it occurs in all cells with nucleus. meiosis only occurs in your reproductive organ, specifically the ovary as well as testicle. The function of mitosis is for growth and repair, while meiosis is to produce gametes for sexual reproduction. Mitosis only involves one cell division that gives rise to two diploid daughter cells. Meiosis, there are two cell division and give rise to four haploid daughter cells. So the daughter cells of mitosis, each of them have 46 chromosomes, while for meiosis, each of the four daughter cells only have 23 chromosomes. Similar to what I have mentioned, the daughter cell of mitosis have the same number of chromosomes as parents. Meiosis only have half. Mitosis, the daughter cells are genetically identical, while meiosis give rise to genetic variation. Homologous chromosomes do not pair up in mitosis, but homologous chromosomes pair up in prophase 1 of meiosis. And lastly, crossing over occurs in mitosis but not in meiosis. Lastly, you have to take note that fertilization will give rise to genetic variation as well, because each of the sperm, they are not genetically identical. Each of the egg, they are also not genetically identical. So depending on which sperm fertilize which egg, the offspring will have different genetic makeup. So this will be all for the chapter of cell division.